All right, welcome back. This is the second half of Chapter 10, where we're going to talk primarily about genetic changes that can occur in the influenza virus, some of those being historical in terms of the fact that they've caused notable disease. And then at the end, I want to talk briefly about some of the very pathogenic bird influenzas that are lurking out there and pose a major risk to human health. All right, so we've already discussed the fact in other chapters that RNA viruses are very prone to mutation because the RNA-dependent RNA polymerase that replicates the RNA genome does not have proofreading capability, and so you will therefore accumulate mutations at a pretty rapid clip. So these kind of point mutations that you would get during RNA replication will lead to what's called genetic drift. So these are just going to be small point mutations that can result in the creation of new influenza strains. And this is going to be in contrast to new subtypes, which I'll talk about in a minute. But if any of these point mutations happen to occur in the HA or the NA proteins, which, as we mentioned, are the major proteins recognized by the human immune system, then you may end up in a situation where a flu virus that you previously had immunity to has now escaped that immunity due to the fact that it has become a new strain and acquired these changes. So in larger changes would be considered antigenic shifts, and this will primarily happen through this process of reassortment, which again we sort of touched on in an earlier chapter, but this can create whole new subtypes of influenza virus where you have new combinations of the H and N variants brought together. And this would happen when you have two different subtypes of influenza that have co-infected a single cell. And it really only happens because of the segmented nature of the influenza genome. So you've got your two subtypes, right, both infecting this cell at the same time, both going through their replication cycle. And when it comes time for assembling new virions, we discuss the fact, right, this is happening at the cell surface. The ribonucleic acid particles are being attracted to the cell surface and will bud off into these new viral particles. And so every new virion is going to get one of each genome segment, but because we have two different subtypes here in the same cell, we may end up grabbing segments from both of those different subtypes and combining them together, as you see here. So now you've got this larger scale change, and if we grab one of the nucleic acid molecules, again, that codes for either HA or NA proteins, then we've got this brand new combination that may not have existed in the world previously. And so this is a big concern because a lot of the different H or N subtypes have never circulated in the human population, meaning humans would have no immunity to these combinations. Our immune systems have never seen them before, but they are out there in the animal world. In particular in birds, as we'll talk about here in a few minutes. So there have been a number of these larger shifts over the years that have, as I alluded to, have been very significant in terms of human disease. Of course, the big one being the 1918, sometimes called the Spanish flu, that killed millions of people around the world in the course of a year. And interestingly, there's some local ties here, so it is believed that this outbreak may have actually started at what is now Fort Riley in Kansas in early spring of 1918. And you've probably heard a fair amount about the 1918 pandemic because of some similarities with the current COVID-19 pandemic, but it was on such a huge scale that hospitals were rapidly overwhelmed, and you were having to create, makeshift wards and gymnasiums and basically warehouses. Any available space was basically being used to house patients. And of course, you got some of the typical influenza symptoms at a very severe level, very high fever, bad cough, etc. But most of the deaths were being caused by bacterial pneumonia or just overwhelming buildup of fluid in the lungs. And so we had a fatality rate of a little over 2% with the 1918 influenza. And keep in mind, at that time, right, this was pre-antibiotic era, so a lot of those deaths due to bacterial pneumonia might have been prevented had they had antibiotics at that time. And I throw this number out there, it used to be completely unthinkable. So during that pandemic, about 900 people died in New York City in one day. I always thought we would never see anything like that again, but of course we have seen something like that again in the current pandemic, and I've seen a number of articles really comparing, especially the intense outbreak in the early spring with COVID in New York City, really mirrored pretty closely what was going on with the 1918 influenza. So that was a scary one, and there's been a big attempt over the years to try to piece together what exactly was the 1918 influenza strain, a little bit of detective work, and best evidence suggests that it was an influenza A, it was an H1N1, basically a avian-like influenza that moved over into humans, may have moved from birds to pigs and then to humans. But both the HA and the NA were coming from bird flu. So again, one of these antigenic shifts that we just talked about, creating this virus that humans had no immunity to, immune systems had never encountered before. And another big problem with the 1918 pandemic, because of that newness to the immune system, we see something quite similar to what's going on with COVID, where you can get cytokine storms in some younger, healthier patients. I think it was happening at an even higher level with the 1918 influenza. But you probably remember cytokines, right, are these very potent immune signaling molecules that are activated early in an infection, and normally they would just be turned on for a short time to get the immune system geared up and running and then shut off. But in these cytokine storms, right, they become hyperactive and end up basically causing the immune system to attack your own body. So in a normal flu epidemic, pandemic, you get a U-shaped mortality curve, meaning, the deaths are happening, this is the dotted line here, deaths are happening in the very young and the very old. Whereas in the 1918 influenza, you got a striking W-shaped mortality curve. So the very young, very old were still dying, but you were also having a large number of deaths in young adults, which was unusual, and that was primarily attributed to this cytokine storm effect. So those with maybe the healthiest immune systems, right, those were more prone to being drug into that storm-like state. So this pandemic was so dramatic, so widespread that overall life expectancy dropped by 12 years in that 1 year alone in 1918, just because of the influenza virus. And I won't go through all these, but I just wanted to point out, right, there have been some other medically significant, quote-unquote, historic antigenic shifts over the years. So creation of H2N2, H3N2, another H1N1, kind of a reemergence of bird flu, etc. And these have all caused significant numbers of deaths. And so that kind of takes us into this idea of the bird flu. sometimes called HPAIs for highly pathogenic avian influenzas. But as I mentioned, right, a lot of these H subtypes and N subtypes are circulating freely in birds and sometimes in bats or pigs or other animals. In certain cases, we would call these epizootic, right? So widespread in animal populations, and whereas relatively few of these have spread in human populations. There's the H strains, there's the N strains again. And so if we think about these avian influenzas being able to infect human hosts, for them to really efficiently spread in humans, they would have to acquire the ability to establish that infection, which of course means attachment primarily. And you might remember that human influenza viruses like to attach to this alpha 2,6 linkage between the sialic acid and galactose molecules that are on these host cell surface proteins, right, in the respiratory tract, whereas the bird flus prefer this alpha 2,3 linkage. And so because of that distinction, most bird flus don't efficiently bind to that human receptor, so it's much more difficult for them to just establish an infection unless you are maybe directly exposed to a very high dose by direct interaction with an infected bird. But what we have seen is in the past 20, 25 years, we've seen some of these bird flus be directly transmitted to humans and cause very severe respiratory syndrome and even death. So this really first came on the radar in the late 90s in Hong Kong. We had about 600 cases of what is now known to be a H5N1 virus, and about 60% of those 600 cases died, so that's a pretty staggering mortality rate there. And almost all of these were thought to be direct bird to human transmission rather than human to human. So really the concern is if these strains acquired the mutations that gave them the ability to easily transmit human to human, right, we could have a very devastating pandemic, something potentially much worse than COVID or the 1918 Spanish flu, right? So that is definitely a concern. And one of the reasons researchers think we may have this extremely high mortality rate in some of these avian influenzas goes back to that idea of their preference for that alpha 2,3 linkage, which, as I suggested, is not really present in most of our respiratory tract. However, this one group of cells pretty deep in the bronchioles, the non-ciliated cuboidal epithelium, do tend to have that 2,3 linkage. So, again, if you get a high enough dosage that the virus is reaching those cells, it may be able to establish that infection, and it's establishing it from the get-go deep in the lung, and so that may contribute to the severity. Just another example here, 2013, there was a new triple reassortment, we would call it, where genome segments were taken from influenza virus from domestic ducks, from wild birds, from domestic poultry, and due to one of these co-infection scenarios, grabbing parts of all three influenza viruses in that case. And again, we had new H and N combinations that humans had not seen. And so, again, most of these are coming through direct contact with those birds, right? Often in either in a farm setting or in a live market type setting, which tends to be more unique to Asia, right? You don't see a lot of live animal markets in the West, typically speaking. A little bit lower mortality rate in this case, but still much, much higher than COVID or even the 1918 influenza at 30%. So anyway, I'll stop there, but just keep in mind these bird flus are out there, but the good news is researchers are keeping an eye on them. So um, hopefully if they do acquire that ability to more easily transmit to humans, we would be prepared and be able to head them off at the pass.